Hi there, and thanks for tuning in. I'm here on the U.S. National Library of Medicine to read to you guys about this study that was done on the effects of marijuana and nicotine on memory performance and hippocampal volume. This is who it was made from. This is a direct reading from the author manuscript. I have not contributed whatsoever to this project other than this video, and that's not even by them. This is all me. Fair use is for educational purposes. I am not planning on making a profit off of this video. Combined use of marijuana, MJ, and tobacco is highly prevalent in today's population. Individual use of either substance is linked to structural brain changes and altered cognitive function, especially with consistent reports of hippocampal volume deficits and poorer memory performance. However, the combined effects of MJ and tobacco on hippocampal structure and on learning and memory performances remain unknown. In this study, we examined both the individual and combined effects of MJ and tobacco on hippocampal volumes and memory performance in 4 groups of adults taken from 2 larger studies. MJ users, N equals 36, and nicotine only, Nic only, N equals 19, combined marijuana and nicotine users, MJ plus Nic, N equals 19, and only using healthy controls, N equals 16. Total bilateral hippocampal volumes and memory performance, WMS3, logical memory, were compared across groups controlling for total brain size and recent alcohol use. Results found MJ and MJ plus Nic groups had smaller total hippocampal volumes compared to Nic only and controls. No significant difference between groups was found between immediate and delayed story recall. However, the control showed a trend for larger hippocampal volumes being associated with better memory scores, while MJ plus Nic users showed a unique inversion, whereby smaller hippocampal volume was associated with better memory. Overall, results suggest abnormalities in the brain behavior relationships underlying memory processes with combined use of marijuana and nicotine use. Further research will need to address these complex interactions between MJ and nicotine. Keywords. Introduction. Marijuana, MJ, and tobacco products remain 2 of the most widely used substances worldwide. In the U.S., combined use of both substances is upwards 60 to 70% in MJ users and more than 5 times as likely as measured by past month use in tobacco users. Moreover, in some countries, smoked MJ joints are extremely exclusively mixed with tobacco. Despite the widespread prevalence of MJ and tobacco co-use, interactive effects of marijuana and nicotine are scantily characterized in the existing literature and lacking direct comparisons of separate MJ only, nicotine only, and combined uses, MJ and nicotine, is a limitation in most studies of marijuana use. Individually, MJ and tobacco are associated with changes to brain structure and function. Structural neuroimaging studies in MJ users have indicated that volumes of several brain areas are smaller in heavy MJ users, especially in areas enriched with cannabinoid type 1 CB1 receptors, such as medial temporal lobe structures. Of these structures, hippocampus appears to be particularly sensitive to heavy marijuana use. Delta 9 tetrahydrocannabinol THC, the primary psychoactive component in marijuana, which binds to CB1 receptors, is associated with cell shrinkage and damage to DNA strands and THC-treated hippocampal neuron cultures. The association with these alterations, such as smaller hippocampal volume with greater lifetime duration of use and cumulative amounts, as well as with recent use, suggests these changes are consequences of exposure to MJ. A recent study by Smith and colleagues examined the interaction between cannabis use and schizophrenia on hippocampal morphology and found a main effect of cannabis use such that altered hippocampal shape was found in both cannabis users with and without schizophrenia. Moreover, these hippocampal differences were related to poorer episodic memory performance, emphasizing the relationship between hippocampal morphology and memory. Taken together, smaller brain volumes in MJ users may reflect potential neurotoxic influence of exogenous cannabinoid exposures. Relative to MJ, less is known about the structural brain changes specific to chronic nicotine use. However, existing studies report lower gray matter densities across widespread areas, example, prefrontal cortex, cingulate gyrus, parietal lobe, cerebellum, thalamus, striatum, and medial temporal lobe in tobacco users. Animal models of rats exposed to nicotine showed reduced cell numbers, increased markers of apoptosis, and alterations in synaptic activity in these regions. These regions express dense levels of acetylcholine receptors that are primary binding targets for nicotine, which further supports the potential for nicotine-related brain changes. Thus, it is likely that, similar to MJ's effects, reported morphometric changes result from nicotine-related neurotoxicity. In addition to structural changes, MJ and tobacco have also been individually associated with declines in cognitive function. Existing studies suggest that tobacco use is associated with impaired working memory, attention, and verbal abilities that map to brain structures that undergo changes due to tobacco use. Example, frontal and parietal cortices, striatum, and hippocampus. In terms of MJ's effects on cognition, studies have reported widespread deficits across various domains such as memory, attention, learning, that are dependent on CB1 receptor activation. However, deficits in working memory appear to be the most consistent. While individual studies provide evidence for neurocognitive consequences of MJ and nicotine use, the independent drug effects may not generalize to the context of combined use. Interactions between the two substances have been described at cellular level, wherein CB1 and nicotine NACH receptors are densely co-localized in hippocampal regions, and both are involved in a diverse set of modulatory processes. For review, see Viveros et al. 2006. That's the source. For example, chronic nicotine treatment in rats resulted in altered endocannabinoid levels in the brain. There's also a pharmacological evidence that cannabinoids alter nicotinic NACH receptor response. Moreover, Valgent Mitchell noticed altered fear withdrawal and tolerance behaviors in rats co-treated with THC and nicotine, suggesting functional biochemical interactions. Taken together, there is convergent evidence from human, animal, and pharmacological studies supporting the potential for additional consequences on the integrity of hippocampal structure and function with combined MJ and nicotine use. However, to date, this has not been yet directly examined. In this study, we aimed to characterize the differential and combined impact of marijuana MJ and nicotine NIC on hippocampal morphometry and memory function among marijuana-only users, nicotine-only users, and comorbid marijuana and nicotine users, MJ plus NIC, with a non-using comparison control group. As a primary aim, we compared groups on hippocampal volume to then further characterize any difference found in hippocampal volumes. We also compared groups on memory performance and examined relationship between morphometry, memory, and substance use patterns. Given findings from existing literature, we anticipated that MJ and nicotine individually and in combination would be associated with smaller hippocampal volumes and poorer memory scores that are inversely related to substance use patterns. Methods and materials. Materials and methods. Participants were recruited through flyers and advertisements in Albuquerque, New Mexico metro area. The community subsample used for this study originated from two larger studies conducted at the University of New Mexico, UNM. Informed consent was provided by all of the participants in accordance to the Institutional Review Board, IRB, at UNM. Participants were compensated for their time. To be eligible for the study, all individuals had to meet the following criteria. A. Be between the ages of 18 and 50. B. Be right-handed. C. Have no magnetic resonance imaging, MRI, contradictions. Example, no metallic implants, pregnancy, claustrophobia, etc. D. Have no symptoms of psychosis via SCID psychosis screen. And E. Be fluent in both oral and written English. Further, individuals with fewer than 10 years of education, IQs less than 75, or illicit drug use other than marijuana were excluded from our sample. We were interested in differences resulting from regular heavy marijuana and nicotine use rather than from recreational marijuana and nicotine use. To that end, marijuana users were also required to report using marijuana verified by urinalysis